Hello everyone. So this is the part 3 of our video series based on CRISPR Cas9 gene editing technology, how to design your project. Although this part 3 was supposed to be how to design your sgRNA based on the gene of your interest, but many of my friends they suggested it would be more better to talk about those terminologies which have been used in part 1 of this series and some other terminologies also which are very important to learn before moving toward your project. So let's talk about it. The very first and important one need to know what CRISPR is. CRISPR is the abbreviation of clustered, regularly interspaced, short palindromic repeats. So this is nothing else, just a region in bacterial genome. But this is repeating after some time, after some spaces. In what is PAM? PAM is protospacer adjacent motif. The CRISPR and the PAM, these are the sequences on the genome of the bacteria. Why PAM is so important? Because PAM is necessary for Cas9 to bind to the target DNA. The PAM will show the place to the Cas9 where it needs to show its action. The PAM is very important in this case because the Cas9 nuclease enzyme will be guided through the single guided RNA, which we will talk about later, to this specific site, and only this site will be cleaved. So it is very important, and this PAM is present just immediately to the CRISPR sequence. What is PAM? It's uh, generally, if we talk, that this is NGG, 3 nucleotides. N can be anything like ATGC, and the two guanines are very important. Now, this PAM has been modified into many formats. Uh, I don't want to go for that explanation. The next is Cas. Cas is the CRISPR associated protein. The Cas9 is a nuclease, endonuclease, which is active enzyme for type 2 CRISPR system. There are like type 1, type 2 systems of the CRISPR. This is in fact the adaptive immune system of the bacteria when viruses attack on these bacteria. So these enzymes, they cleave the genome of the virus and cut it into pieces, and then they can survive. Next, what is CRISPR A? CRISPR A is CRISPR activation. You have to use dCas9 or dCas9 activator with sgRNA for what purpose? To increase the transcription of the target gene. We will talk about it in more details. Uh, I will also make a single video based on CRISPR A and CRISPR I. These are very important. For now, you just understand that this is the CRISPR-A we can use for the purpose to overexpress the gene of our interest. Next is CRISPR-I. What is CRISPR-I? CRISPR-I is CRISPR interference, and this is totally opposite to CRISPR-A. In this case, dead Cas9 or dead Cas9 repressor is being used for the purpose to repress or decrease the transcription of your target gene. In CRISPR activation, CRISPR-A, we need to use the dCas9 and the activator. For activator, we have to use VP64 domain. And here, for CRISPR interference, we need to use a repressor. That repressor is CREB domain. I will also make a single video based on CRISPR-I to explain it in more details. What is cut? Simply, cut is like a bite. Yes, bite on the strand of DNA, double strands. So, cut means we cut down the double strand of DNA into pieces. Next is dead Cas9, dCas9. These are the formats of the Cas9. One is wild Cas9 that can cause double strand breaks, means can cleave both strands of the DNA, both backbones of the DNA. But dead Cas9 is an enzymatically inactive form of Cas9. It means the nuclease activity of the Cas9 has been deactivated. So now this Cas9, dead Cas9 cannot make the DSB, double strand break. If it cannot be used to cut the DNA strands, then why we are using it? Because we have to to deliver this machinery to the targeted region. So that's why Cas9 is being used. The next is DSP, double strand break. Double strand break, you know that the DNA composed of double strands, and if we make cut on one strand and other strand, so that is called double strand breaks. We can use Cas9, the wild Cas9, or we can use the two Cas9 nicases of targeting opposite strands. Uh, this one is a little complicated for beginners, so if you are just moving toward the CRISPR, so you can just ignore it. Later on you will understand it in very best format, in very best way. Dual nicase or double nick, this is what I was just talking about. In this method you create a break, a cut on one strand, and the other cut is not just where you made cut, like just opposite to the same cut on the same strand, but it's in uh, different positions. And for this purpose you have to use two different single guided RNAs, simply we can say the guide RNAs, to bring this machinery to that place. We will talk about it in quite details later. The guide RNA. This is the very important component of the CRISPR-Cas9 gene editing system. This is the synthetic fusion of the endogenous bacterial CRISPR RNA and trans-activating CRISPR RNA. We will talk about it in more details and you will understand it very well. For now, you just imagine that the guide RNA is the complementary strand just to the CRISPR. Again, I'm repeating, guide RNA is the complementary strand just to the CRISPR, the segment of DNA where do you want to make a cut. So you just, for this time, you just imagine that this is the complementary segment, the complementary strand just to that CRISPR. Later on we will talk about in more detail and you will understand. Both targeting specificity and scaffolding binding ability for Cas9 nuclease does not exist in nature, also referred as single guide RNA. Mostly I will use the term single guide RNA because I'm habitual to this one. This is very easy to understand. Don't be in stress. Just go to next. If you don't understand it well, so just go to next step. Guide RNA scaffold sequence. This is the sequence of DNA which is responsible for Cas9 binding, and this is not included in the 20 nucleotide base pair spacer or the targeting sequence that have been to be used to guide the Cas9 to the target DNA. You can see I have shown it from the ED gene, and it is very easy. We will talk about it quite detail. Guide RNA targeting sequence. This is the 20 nucleotide that precede the PAM sequence in the genomic DNA. Where do you want to make a cut? Where do you want to break the strands of the DNA? That is your targeting sequence for guide RNA. Just now I just talked about it, that the guide RNA is complementary to your CRISPR spaces, CRISPR sequences. So that CRISPR sequence is your target sequence. HDR, homology directed repair, a DNA repair mechanism that uses template to repair nicks or double strand breaks. You might know about it in quite detail. Indels means insertions and deletions. When you do some disruption with that gene and that gene is disrupted, then after like homology directed repair or NHEJ, the repair of the DNA system, and then the ORF of that gene is disturbed, so that is called indel. You can make premature stop codons by doing this. This is mostly used in base editing. NHEJ, non-homologous engineering. This is a DNA repair system. It's a very common phenomenon and you might know about it. Nick. Nick means you just make a cut on a single strand of double-stranded DNA. This is normally repaired by homology directed repair, and this is mostly used in the single base editing. Cas9 that has one of the two nucleases domains inactivated can either be ROVC, this is one endonuclease domain, or HNH domain, this is another uh, endonuclease domain. So if one of these domain is deactivated, so that Cas9 will make a single cut on that double-stranded DNA. Off-target effects means because the genome of that organism is very big. For example, if we talk about the mice, so the mice genome is very big. You are trying to find the exact location where do you want to disrupt that gene, where do you want to make the cleavage of that desired gene, but it goes to the which places where you don't want to take them. And to avoid from off targets, in very first part of the series of the video, I have talked about like we will authenticate our sgRNA from NCBI blast tool. So we will see to reduce the off targets, and for this purpose we can also reduce the off targets by using the double nicks, but this is a little complicated mechanism and it's also the efficiency is quite low. On target activity, where you want the desired location, exactly it goes to that place. So this is very important, and mostly it goes like that, but off target is still you can say a nightmare. ORF, open reading frame, this is the codons that make up a gene, and if you make some disruption, if you change a single base, so the uh, ORF, the open reading frame of that gene is disturbed, and in this way that gene can be converted even into uh, premature termination. Gene knockout, you permanently disrupt that gene, particular cell or organism. You don't care about that later on you will need this gene in that organism, so you just take it out from that cell or that organism. Universally disruption, you play with that gene in the whole body of that organism. Conditionally disruption, you take that gene to a specific tissue or a specific place, and in this case you need a promoter. That promoter will take this machinery only to that tissue. I'm using some different promoter and that is unpublished, so I cannot talk about it. Inducible disruption of the gene, that means when do you want to play with this gene? For example, you are playing with the gene that is related to the age, and then for example the mice is 1 year old and you want to know the function of the gene, like in 1 year old mice overexpression or down regulation or whatever, so you have to use the inducible disruption. And for inducible disruption, uh, one need to go for the TET system, TET R, TET O, TET A. We will talk about it in details. So friends, in this video I just talked about the very important, very basic terminologies which are, you can say, the backbone for the CRISPR Cas9 gene editing technology. One might know about these terminologies if he or she is planning to design the project and work on it. The next video will be based on the design of sgRNA, and we will use different tools how to design sgRNA. But for now, that's it.